In this short video, we will talk about the ear. Roughly, the ear is divided into three main areas. The external ear, which includes that cartilaginous part that we see, the pinna, as well as the external auditory meatus, and all the way to the tympanic membrane. So it is a skin-lined cavity. The external auditory meatus largely innervated by the auriculotemporal nerve off of V3, as well as, or the third division of the trigeminal nerve, as well as some vagus contributions, particularly on the inferior part of the external auditory meatus and ear canal. The middle ear, its innervation is largely via the glossopharyngeal nerve, which makes sense because that middle ear connects the middle ear. It connects the ear to the nasopharynx. And so the middle ear includes the nasopharynx, the pharyngeal tympanic tube, which recall that it opens into the posterior aspect of the nasopharynx, immediately inferior to the torus tubarius, which is this mucosal outpouching just superior to the soft palate. It also includes the adductus to the mastoid antrum, so opening to these air-filled, mucosal-lined air cells in the mastoid process. It contains the ossicles, the malleus, the incus, the stapes, those bones, those tiny little bones within the middle ear. The internal ear contains the cochlea, which houses the organ for hearing, as well as the utricle and saccule are also a part of this inner ear, as well as the semicircular canals, which are part of the vestibular or balance apparatus, and then all the way to the internal acoustic or internal auditory meatus. You may see both of those terms. When we're talking about the ear, usually we're thinking about this cartilaginous portion that's a part of the external ear, the pinna. And within it, even within those cartilaginous folds, are structures that we need to talk a little bit about. So the helix there is on the rim of the ear, and then the bulge immediately anterior to that is the antihelix. The concha is that inner part of the ear, the cartilaginous ear that we see, and the crust of the ear is here. Interestingly, the tragus is named tragus for this, like a sheep's beard. And so if you've ever seen on elderly gentlemen where they have a little tuft of hair coming from their tragus, is because it is reminiscent of a goat's beard, that it has this name tragus. Immediately opposite that is that antitragus. Now, in terms of sensory to the ear, that posterior part, so posterior to the helix, and including the helix, the innervation is via the lesser occipital nerve, and more inferiorly here on the lobe, the great auricular nerve. Remember, both of those are part of the cervical plexus that we talked about in the neck, moving superiorly from the nerve point of the neck, crossing the sternocleidomastoid partially. And so those nerves will provide innervation to the posterior part of the ear here. More anteriorly, however, The antehelix, for instance, is innervated by parts of the facial nerve, and then the concha here with a auricular branch of the vagus nerve. This most anterior part is innervated by the auriculotemporal nerve and a little bit of vagus as we move deeper, and that extends into this into the external auditory meatus to the ear canal here. Now, in adults, the ear canal here is the most, external part. The external or outer 1/3 is cartilaginous, just like the ear was, but the inner 2/3 are largely bony. Okay, and so in adults, this ear canal adopts this kind of S-shaped structure, as opposed to in children where the ear canal is a lot straighter, and that is thought to contribute to the frequency of infection in little kids. So in adults, however, this S-shaped structure of it means that when your physician needs to look with the otoscope down the ear, it helps to pull it superiorly, pull the ear itself, the pinna of the ear, posterior superiorly, in order to straighten out the ear canal so that you can see it. So I have here written to remember to tell you about otitis externa. And so that really is inflammation or inflammation or infection in the external ear, where the external ear remains wet, and then it can then result in infection, either bacterial or fungal, such that that's what swimmer's ear is. Now, if we continue towards the middle ear, still in the external ear, we see the tympanic membrane. So the external surface of the tympanic membrane is covered in skin, as opposed to the internal surface, which is covered in mucosa. This is a concave, so the tympanic membrane is a concave structure, so it projects into the middle ear, and you can see the bulge from the malleus that is anchored to this most anterior part, this pars flaccida, so this flexible part of the tympanic membrane. By and large, though, the rest of the tympanic membrane is the pars tensa, the tense part that lacks the flexibility of the pars flaccida, which is this this more superior part where the malleus is projecting onto the surface of the tympanic membrane. You can see the knob or umbro, as well as the as well as the handle, the lateral process, and their folds of the pars flaccida as the malleus projects onto the tympanic membrane, making this impression. So if we're viewing the tympanic membrane with our otoscope on an adult where we're pulling posterior superiorly, the light from the otoscope creates this cone of light at about 5 o'clock. That is characteristic of the reflection of the light as you're looking into the external ear. Tubes in your ears or any surgical procedure often happens in this posterior inferior quadrant due to the lack of or the absence of the nerves that are part of the middle ear, you're less likely to encounter them in this posterior inferior quadrant of the tympanic membrane. So here is another view from the otoscope, and then remember that the cone of light here is at the 5 o'clock position. And so we have the umbral, handle, lateral process, and then incus and stapes. You can see some impressions in this pars flaccida part of the tympanic membrane. The pars tensa is here, and if we wanted to put tubes in the ears, we would be in this inferior posterior compartment. So here is that tympanic membrane, but in this image you can start to appreciate the ossicles of the middle ear. The ossicles are these tiny bones. The malleus connects to the tympanic membrane, and then when the sound waves move the tympanic membrane, it then causes the ossicles to move, which cause the incus to move, which cause the stapes to move, and at the membrane at the oval window. And we'll talk a little bit more about that. This middle cavity, this middle ear cavity, is innervated by the glossopharyngeal nerve. And then here is that pharyngotympanic tube or auditory tube that leads towards the posterior nasopharynx. Okay, so this is the tympanic cavity, and then the top part is the epitympanic recess. The adytus here is, so an adytus is a doorway, and so this is a doorway to the mastoid air cells, mastoid antrum. And so this is connected to the mastoid air cells. And remember that this bony part is really embedded in the petrous part of the tympanic, the petrous part of the temporal bone. And I'll show you another image of that as we go along. When we talk about otitis media, we're really talking about an infection in this middle ear cavity, right? It makes sense. That media part is that middle, the middle ear. Oftentimes it is due to this. So in the case of my son Ian, when he was having lots of inner ear infections and he had those adenoids, that connection there is what facilitated the infections in the middle ear. So that big hypertrophy of that pharyngeal tonsil blocking the pharyngotympanic tube in the posterior nasopharynx, then allowing for infection to set up in this middle ear. So this is another view of the middle ear so that you can appreciate its complex architecture. So this blue wall is the membranous wall that contains mostly the tympanic membrane. And then notice your malleus, incus, and stapes, those ossicles running through this middle ear cavity as the stapes then attaches at the oval window. Also in this picture, you can appreciate this chorda tympani. And the chorda tympani is a branch off the facial nerve. Now the facial nerve does not run through the middle ear, but it does bulge on the other side of this membranous labyrinthian wall. So there's a bony wall right here, and you can see a bulge that immediately deep to that is where the facial nerve would be. The facial nerve is going through this facial canal, and along the way it gives off the nerve to the stapedius, which is this little tiny muscle here. Stapedius is this muscle, and when it contracts, it limits the oscillations of the stapes at the oval window, dampening the sound a bit, or at least the oscillations, which then result in decreasing the sound. And so there's a little tiny nerve to the stapedius that comes off the facial nerve that innervates this muscle. Also, the facial nerve gives off this chorda tympani. The chorda tympani goes through this middle ear. You saw it in the infratemporal fossa joining the lingual nerve. And so that's that very same chorda tympani that you saw during that dissection of the infratemporal fossa. The tensor tympani is here. It sits in its own canal superior to the pharyngotympanic tube. And the other part of it, it's cut here. The other part of it is at this wall that is closest to you that we're looking through. Okay, the tensor tympani, when it acts on the malleus, it limits the movement of the tympanic membrane, or the oscillation of the malleus at the tympanic window. Again, tensing that tympanic membrane, decreasing the sound. For chronically loud sounds, it kind of turns it down, right? All right. And so this back wall is the mastoid wall, and the opening, that red part there, is the adductus to the mastoid antrum, the opening to the mastoid antrum, right? Also bulging into this wall is the lateral semicircular canal. It's not in the middle ear, but the bulge from the labyrinthian wall projects into, so there's a bump in the middle ear, a bony bump in the middle ear, that immediately on the other side of that bone is where the semicircular canal projects into. And then you also see a bulge here from the cochlea. And so this, the cochlea is responsible for hearing. And so this bulge is from the turn of that snail-like cochlea, where it projects onto this wall. Going over the wall is the tympanic plexus. And so from the glossopharyngeal nerve, as glossopharyngeal nerve sends a tympanic branch through a structure called the tympanic canaliculus, and the tympanic nerve then supplies this turn of the cochlea, or the tympanic promontory, the promontory on the labyrinthian wall. Now, the tympanic nerve from glossopharyngeal provides the parasympathetics that are a part of this plexus. The sympathetics come from the sympathetic plexus that accompanies the internal carotid artery, which is in this space that we're looking through. And I'll show you another image of that so you can really appreciate the complexity of this area. So this tympanic plexus includes sympathetics that have come from the sympathetic nerves accompanying or surrounding the internal carotid artery, and it contains parasympathetics that originated with the glossopharyngeal nerve that got to this tympanic plexus via the tympanic nerve. From there, the lesser petrosal nerve can then carry these sympathetics, parasympathetics to some other places that we'll talk about later. The bottom wall is the jugular wall because the internal jugular vein is in that space. And then the carotid wall, again, has been removed so that we can look inside the middle ear. So you don't need to know this facial nerve nucleus, but you do need to know that these SVE fibers, special visceral efferent fibers that originated with the facial nerve, travel through the facial canal. That facial canal is the very same facial canal that we saw bulging into the middle ear on the other side of the labyrinthian wall. And along the way in the facial canal, it gives off this nerve to the stapedius, and then the facial nerve exits the stylomastoid foramen. It also gives off that chorda tympani that's shown here. It's not drawn in, but the facial nerve then exits the stylomastoid foramen and then goes to those muscles of facial expression as well as the posterior belly of the digastric, the stylohyoid, and those other muscles of facial expression, those auricularis muscles that would, if we were animals of a different species, you've seen those animals where they move their ears toward the direction of sound, right? Humans don't really do that. And so there's just kind of a remnant of that, that they are less developed, let's say, in humans. Here's that picture again, and I want you to appreciate in this next picture, here's that carotid canal. So in this view, this was the wall that had been removed so we could see the inner ear. But here, this is a different view where you can see that anterior wall, which contains the carotid canal and the pharyngotympanic tube, which leads to the nasopharynx, as well as the bony canal that houses the tensor tympani, which comes through the middle ear and acts on the malleus at the tympanic membrane. You can also see the jugular wall and the jugular foramen here. Remember those cranial nerves that also exit the jugular foramen in that region. Here's our tympanic nerve, which goes through this tympanic canaliculus to get to the promontory, where it then participates in the tympanic plexus. And then in this image, the bulge of the facial, the bony covering of the facial nerve has been removed so that you can appreciate the facial nerve in the facial canal as it gives off its nerve to the stapedius, traveling through the facial canal to get to the stylomastoid foramen. Here's the mastoid process, and the adductus is here. The antrum is here, and that's the opening to these mastoid air cells, these mucosal lined cells inside the mastoid process. Here is an image from your cranial nerve notes. Same thing, but in this picture, what I like about it is that you can see what's been drawn in is this carotid plexus, these sympathetics that accompany the internal carotid artery that participate in this tympanic plexus with the parasympathetics that came from the glossopharyngeal nerve going through the tympanic canaliculus. Okay, and then you can see as that goes on to become the lesser petrosal nerve in this area. So here is one of those cranial nerve charts. Remember, these blue backgrounds are just to give you a little bit more context, although I'll talk more about these different fiber types in the cranial nerves video that will come next. So in this image, I'm only showing you these GVE fibers. You don't need to know the inferior salivary nucleus, but what you do need to know is that glossopharyngeal fibers, preganglionic glossopharyngeal fibers, go through the glossopharyngeal nerve, go through this tympanic branch, which goes through the tympanic canaliculus, go over to this tympanic plexus, and then via the lesser petrosal nerve, these preganglionic parasympathetic fibers then synapse in the optic ganglia, and then postganglionic parasympathetic fibers get distributed to the parotid gland through the auriculotemporal nerve. And remember that the auriculotemporal nerve is a part of the trigeminal nerve. So when we're talking about the ear, we're talking about this bone-lined structure that contains this membrane and fluids within the petrous part of the temporal bone. And that's shown here, as well as how they're oriented, where the cochlea is here, and then the semicircular canals are laterally in this space. So in this image, they've basically taken the otic capsule, so the extra hard part of the bony part of the ear, and only taking this outer cast of it. Remember that it is lined with these membranous and fluid-filled cavities within this hard bony otic capsule within the petrous part of the temporal bone. You can see this snail-shaped cochlea where it takes 2.5 roughly turns in this snail-shaped cochlea, which houses the organ for hearing. And then you also see bulges in the vestibule where the utricle and saccule, which sense acceleration, horizontal and vertical acceleration, where those membranous organs lie, as well as these semicircular canals that are also lined by these that are bony here, but contain membranous and fluid-filled organs that then allow for us to detect position. So these organs of balance are located in these parts on the right. There's also a little bulge here of that facial canal where the facial nerve lies. So here's that same space again. In this image, though, you can appreciate the membranous labyrinth that is within that otic capsule, right? And then the utricle and saccule are shown here, which sense acceleration. And then the semicircle canals are located here, where we can sense or understand positioning. Now, within these cavities, this membranous sac is a substance called endolymph, and the endolymph is shown in this picture here in green. And so there are membranes surrounding or enclosing this endolymph-filled cavity. And then on either side of this are these perilymph fields. Perilymph is a fluid-filled cavities on either side of that. The The cochlea, then these fluid-filled cavity, I told you it takes 2.5 turns as it's forming this shell-like projection. And the center part there is that it rotates around is the bony part is the modiolus. And then the cochlear nerve projects from there after being connected to the membranes that are therein. So this is just another picture of the same structure. Okay. So once the let's just orient ourselves, I guess, a little bit with the structures here, where we have the stapes from the middle ear sitting at the oval window. And the oval window has another membrane in it. And so then as we move to the inside the cochlea, right, so it's unwound in this picture. The middle part here that's in white, this scala media, is the endolymph filled cavity. And then on either side of this is perilymph, right? Like peri is beside. So perilymph cavity. The scala vestibuli and the scala media are separated by the vestibular membrane or Reisner's membrane. And then the scala media and the scala tympani are separated by this basilar membrane. And it's the basilar membrane that contains the organ of Corti, the organ of hearing that converts mechanical movement into nerve impulses so that then we can sense or detect hearing. So if we hear a sound, often it deflects this tympanic membrane, and the movement of the tympanic membrane moves the malleus, which moves the incus, which then moves the stapes at the oval window. This movement then creates a pressure wave that then moves through the cochlea, which deflects these membranes. And these membranes then, particularly the deflection of the basilar membrane, then changes the nerve impulse, and then that then gets transmitted to the cochlear part of the vestibular cochlear nerve. Notice that the scala vestibula and the scala tympani meet at the apex, and the apex is called the helicotrema. I forgot to say that part. All right, so here again is our blow up of our organ of Corti in here's our scala media, here's our basilar membrane. And there are these hair cells which have cilia on them that project into this gelatinous tectorial membrane. When this membrane moves, it then changes it deflects, and that then changes the frequency of the nerve impulse that's sent via this spiral ganglia. So here is what I mean. So before it's deflected here, and notice you have these little hair cells that project into this tectorial membrane, and then normally, and then once that basilar membrane gets deflected, once it moves due to that pressure wave, it changes the direction of the interaction of these hair cells on this tectorial membrane, which then leads to the stimulation of this nerve, this cochlear nerve, which is responsible for us then being able to sense hearing. The basilar membrane is, at its base, is thin and quite flexible, and then as it gets to its apex all the way to the helical trema, then it gets thicker and it produces the lower frequency sounds at the apex if the wave makes it to the apex, and then higher frequency sounds are going to be detected more towards the base, so more proximally, I guess is a better way to kind of describe that. So this basilar membrane gets thicker and wider as it goes towards the apex of the cochlea. So in this picture, you can appreciate this basilar membrane, and if the wave propagates closer towards the base of the cochlea, then it's a high frequency sound, and if it continues on towards the apex, it produces this low frequency sound. All right, so the next thing I want to talk about is actually the utricle and saccule. Notice that the scala media in its endolymph fields, this endolymph field membranous cavity is continuous with the saccule, and the saccule is continuous with the utricle. The utricle senses horizontal acceleration as opposed to the saccule, which senses vertical acceleration. Utricle and saccule are part of the vestibular apparatus. So in addition to the semicircular canals, it allows us to understand our positioning, our body's positioning, for instance, or detect positioning in space. In the case of the utricle and saccule, that sensation of vertical or horizontal movement. What you can't see here is the endolymph sac, which is posterior, I guess, to this, where the endolymph is reabsorbed after having filled the scala media, the utricle and saccule, as well as the semicircular canals or endolymph field. And so that brings us here. So at the top here, you can see the semicircular canals. They are oriented in three different directions. Those semicircular canals have this membranous endolymph filled space that I've indicated with the E, and on either side of the membranes are perilymph filled spaces. Within the endolymph filled space, so here's a blow up, there is a structure called the cupula, and that contains this this part of this crista ampullaris, okay, crista ampullaris, and then this gelatinous part here is called the cupula. So there is a difference in the specific density, for instance, of the cupula relative to the endolymph, and so when we change the positioning of, let's say, our head, it deflects the hair cells here that project into the cupula, and so those hair cells contain these cilia, particularly this extra long cilia called the kinocilium, when the deflection is of the hair cells is toward the kinocilium, it then stimulates the nerve impulse to then go through this vestibular ganglia, and then the nerve impulse, the nerve then goes to the brain, so we can then detect balance. So we can then use that information to detect balance. The same is true if it deflects away from the kinocilium. It inhibits this signal, and it's the reading, so to speak, of the nerve impulses that are sent by these vestibular ganglia that then result in us being able to detect or have balance with these three different semicircular canals that are, you'll see those in the lab. There's a big ear model that you can then appreciate the direction of the semicircular canals. Here's a picture of the utricle and saccule. There's also these kinocilium, right? This extra long cilia. What's different about that space, the utricle and saccule, is that they have these otoliths, which are calcium carbonate crystals that create this otolithic membrane. So when there is movement, because of the difference in density of the otolithic membrane, there's then a lag. If there's movement, the endolymph and the otolithic membrane are deflected and there's a difference in the deflection, and that allows us to then determine if we're moving horizontally or vertically, for instance. And so that's the otoliths within the utricle and saccule. All right, so those organs of the vestibular apparatus, here's a utricle, here's our saccule. They contain these macula, and the macula are where the kinocilium and the otolithic membrane lie. Okay, and so it's the deflection of those hair cells that is integrated into this impulse that's then sit through this vestibular ganglion to form the vestibular part of the vestibular cochlear nerve. Here's that endolymph sac that I was telling you about, where the endolymph is reabsorbed. Okay. And then the cochlea here is a bit unwound, and the cochlea, then the deflection of the hair cells on at the tectoral membrane then controls the nerve impulse that's sent via these spiral ganglia to the cochlear part of the vestibular cochlear nerve. Here they combine and go to the internal acoustic meatus before going to the brainstem. All right, so the last thing is the Rene test and the Weber test. I posted a small video on Moodle, and we have some tuning forks so that you can play with those tests in the lab. And so I would encourage you to take advantage of that opportunity to try to understand.